This is the lecture for biotechnology for a lecture that would have been held on Friday, the 11th of January. And I'm starting in where we left off in chapter 1. And we were talking about the different types of biotechnologies, and we left off on medical biotechnology. And this is involved in obviously the whole spectrum of human medicine, being able to prevent different types of diseases, diagnostics, where you're diagnosing disease, and then also treatment. And at the crux of this is the Human Genome Project. And we talked about this earlier on Wednesday last week, where the entire genome was sequenced by about 2003. The project was supposed to take 20 years, and it only took 5 years to sequence the entire human genome. And this is not only gene therapy, but a lot of information in terms of different diseases that are associated with genetics is now available, and also different susceptibilities for different things like cancers, heart disease, high blood pressure, things like that have been elucidated. So you can prevent these by knowing which patients are much more susceptible to those diseases and then intervening before they get those diseases. And then finally, stem cell technologies. And stem cells have been grown in the laboratory. They can be adult or embryonic. And then stem cells are undifferentiated cells. These are cells that haven't differentiated into, say, different tissues or organs, but then they can be stimulated by different types of chemicals to develop specific types of tissues that are needed for transplant. You can use stem cells. You can seed stem cells on skin grafts. You can seed stem cells to do liver transplants and organ transplants. And although this is a very expensive technology, it's also very promising. And stem cells are currently used for diabetes to regenerate pancreas tissue. Also, they're starting to use this. This is more experimental, but they're starting to use this for spinal cord injuries to repair the spinal cord and reintroduce some function into the spinal cord. And obviously, genes are in the headlines. It's very easy to do genetic sequencing these days. And it's very easy to well, I guess it's not as easy to ascribe a particular gene to a particular type of function or a particular type of genetic disorder. But anyhow, you see a lot of this information on the news. Okay. Another type of biotechnology, and in discussing the regulatory environment this semester, we will talk about these roles in biotechnology. But one in a commercial aspect is quality assurance. And that is making sure that the final quality of a product is regulated by monitoring all activities that go into that product to make sure that you've got sufficient efficacy of the product, sufficient purity, and that there are no contaminations that are going to cause problems. Part of quality assurance is quality control. And quality control is more when you take, let's say, something that's being produced and you test it in a laboratory. A quality control laboratory assures that a product is uniform and the product is appropriate to all the different standards. If you're making insulin, you want to make sure that it lowers blood sugar. That you are able to combat high blood sugar, which is really bad for you, and that the insulin is not contaminated. So quality control is when you take that sample of insulin that's being produced in a manufacturing facility and assuring that it's meeting all of its product standards. And we should care about this. Quality assurance and quality control are a big part of biotechnology, because you're using this for a lot of different products, for a lot of different applications, including things like human health. And if we don't have rigorous standards, then you're going to start to produce drugs and biologics that are going to make people sick. And so a consumer needs to be interested in the safety of the product that they're using, the effectiveness of the product that they're using, and also these are quite expensive. So you don't want to buy a batch of insulin for $5,000 that is not going to work and could, say, worse off, stimulate your immune system because it thinks it's a foreign invader. And if we look at medical biotechnology, the Human Genome Project was really the biggest advance, because now we have a map. We thought, and I can say we because I remember when the human genome was being sequenced, we thought this was going to end all disease and all types of cancers, and just improve vastly the life of humankind, but it did not. And one of the reasons why is the Human Genome Project only gave us the A's, G's, T's, and C's. So it unlocked the sequences of the genes, but there's still a lot of research on the function of the genes that were sequenced and the structure of the genes and how you can manipulate those genes. That research is ongoing. And so there is promise for the future. But when we talk about the Human Genome Project, we also talk in the future, even though the official project ended in 2003, because the human genome is still being unlocked in terms of the function of human genes and the things that regulate those genes. And then the proteome, genome, it means all of the genetic material in a given cell or a given organism. The proteome is all the proteins and the proteins that are stimulated under different conditions in a human cell. And this is also very important. The proteome is more difficult to unlock because the proteome is dependent on a variety of factors. Where our genetic material is fixed, the proteins that are in our cells are variable depending on the different condition of the host. Say if somebody has cancer, they have different proteins in their cells than somebody that doesn't have cancer. And so this is not a fixed target, but it is good to understand how the proteome varies under different conditions for humans. We also have what are called single nucleotide polymorphisms, or SNPs. And if you haven't heard of SNPs before, these are just single DNA-based mutations that you see in the human population. These different SNPs mean that the genes are going to function slightly differently. So these mutations will vary from individual to individual. They're not necessarily mutations that will make you sick, but they are mutations that will give you susceptibility to different type of syndromes like heart disease or stroke or breast cancer. And it's important to note these changes because these individuals are going to react differently to different medicines. They'll have different characteristics. In some cases, a single mutation will cause a genetic disease like sickle cell anemia, but mostly this identifies genes that create greater levels of susceptibility for medical conditions, including everything that is listed here. And even behavioral, even psychiatric illnesses, you'll see certain SNPs that are associated with more psychiatric illnesses. And so medications are being developed for individuals that have these particular SNPs. Okay, for breast cancer, the SNPs that are actually very, very indicative of breast cancer are in BRCA1 and BRCA2 genes. And it turns out that depending on what your genotype for BRCA1 and BRCA2 are, it could mean that your chance of getting cancer in your lifetime could be as high as 80 or 90%. And that's where people are having things like elective radical mastectomies. Some are having hysterectomies because they're highly susceptible to these different cancers. And so if you don't have that particular tissue, if you don't have breast tissue, then you're not going to get breast cancer. Okay. And you see the consequences of different SNPs. If you look at person one, and person two, person one and person two, both are producing a normal protein because the mutation in person two is what's called a silent mutation. It has no negative effect. But you can have a mutation, you can have a SNP, like if we take the G that was replaced with T and then replace it with A, that could create a non-functional protein. So certain SNPs, they don't mean anything. They're just silent. They still code for the same amino acid, but other SNPs will code for different amino acids and they'll lead to a non-functional protein. And again, based on this figure, the reason why the person two didn't develop a genetic disease is because the mutation was silent. When we say that the mutation is silent, it means that the codon that mutation was involved with still codes the same amino acid, even though that particular nucleotide is different. And there's a lot of amino acids, in fact, almost all amino acids except for methionine are encoded by multiple codons. And so there are variations that will give you the same amino acid. And as far as the body's concerned, the protein will be the same, so the person doesn't get the genetic disease. And in order to test a person's DNA for many SNPs, you can do sequencing analysis. A lot of times this is done in microarrays, and you isolate the DNA of the patient, and you apply this sample to a microarray that has many different genes with SNPs in them, and you watch the DNA hybridize. Hybridize is when the strand of the patient's DNA will directly link up with the strand of the sample DNA. And you compare the patterns of DNA binding between the patient's DNA and the DNA on the microarray. And depending how it binds the DNA in the microarray, that will reveal the patient's SNP. So you don't have to do a direct sequence analysis. You can just do this with a genetic array. Okay, in terms of how this is going to change our lives in the future, this is actually the present now. Pharmacogenomics means that certain drugs are given to certain people based on their genotype. So we can tailor drug therapy and treatment based on the genetic profile of the patient. Even myself, I receive certain drugs because I've had a genetic analysis that shows that I'm susceptible to different types of diseases. And so I am practicing pharmacogenomics. And this is just done with microarray analysis. It tells the patient what SNPs they have, and then depending on their SNP profile, they will react differently to different medicines. Also, another type of omics is metabolomics, and this is just a snapshot of the small chemical metabolites that are produced in a cell. This would be small molecules like glucose, cholesterol, ATP, growth factors, and signaling molecules, and this will be able to distinguish whether the patient has a disease or if they have a normal situation. And so it's used for disease diagnosis, and it's also used for the overall condition of the health of the patient. Another type of technology is nanotechnology, and this is just a lipid nanoparticle here This lipid particle is just like a big cell, and within the cell is the therapeutic drug. And this nanoparticle will break apart only when it encounters a cancer cell. And so when it encounters a cancer cell, the therapeutic will be released. And, as you know, and Hillary, as you know, doing chemo, these drugs are very, very difficult on the body. And this lipid nanoparticle will deliver the toxic drug to the tumor itself, and it will save the rest of the body. Okay, so you won't have these systemic side effects because of having something that's very toxic to the body, all over the body, but it will target the tumor itself. Also, there are some gene therapies that are on the market. Gene therapy technology is becoming much more prevalent. And this is where you replace normal genes or defective genes with normal copies of the genes. And this is still, there's a lot of research that needs to be done, a lot of barriers that have to be overcome. And some of the obstacles are how do you deliver a normal gene to all the genes in the body? And what are the long-term effects of introducing extra genes to humans? Sometimes the immune system will not recognize the correct gene sequence because in the patient that has a defective gene, they've never seen that sequence. So the immune system will attack the normal gene. So it's important to know the long-term effects, whether the genes will be rejected or whether they can be maintained in the body. And then also what's being done to make sure that the gene, which is encoding a protein, is actually being transcribed and translated. Another type of technology which blocks gene translation is what's called small interfering RNA. And this silences genes. There are certain genes that if they produce too much protein, they will create disease states like rheumatoid arthritis or certain autoimmune diseases. And by providing a small piece of RNA that is complementary to that protein sequence, then you can block the production of the protein, lower the production of the protein, and stop the disease. Okay, and then back to stem cells. Obviously, you've heard this before, stem cells are immature. They haven't differentiated, and as they grow and divide in the body, then they differentiate and they become different cell types. Okay, they're very useful because by growing stem cells in the laboratory, you can regenerate lots of different types of tissues. And most stem cells are embryonic stem cells. They come from human embryos. And this is very controversial because of life issues, determining when the beginning of life is, and they involve the death of an embryo. But you can get stem cells from adult cells. We produce stem cells all the time as adults, and so these stem cells can grow in the cells of grow into the cells of interest. They can be harvested directly from the patient's body, which means that there's no chance of rejection, and then they can be redifferentiated to regenerate organs and tissues. So we start out with an embryonic stem cell. If it can be differentiated to make many, many different types of tissues. An embryonic stem cell has not committed itself to any type of cell. Okay, so it can grow literally any cell in the body. An adult stem cell, I don't have a slide for this, but an adult stem cell, usually if you extract an adult stem cell from like nasal tissue, it's going to make a nose, or if you get it from liver tissue, it's going to make liver. So it's not completely totipotent, but it's pluripotent. It can make the tissue or organ from where it was extracted. Okay, and then how will medical technology change our lives in the year ahead? Regenerative medicine, where you can regenerate organs and tissue. And genetically modifying stem cells of patients to treat genetic diseases. And in the future, we'll be able to isolate adult stem cells from the patient with the disorder, uh, genetically modify these cells with gene therapy so you correct the genetic defect. This can be done just by providing the correct gene, or you could do it by repairing the gene with CRISPR technology. And then you reinsert the cells into the same patient, and then that helps them combat the disease. Okay, now switching over, we'll talk about the biotechnology workforce. And if you look at biotechnology, right now it generates, this is an older figure, it's probably more like 100 billion worldwide, and more like over 60 billion in the United States. And you see in California, we have the vast majority of the biotechnology companies, over 350 different biotechnology companies. And pharmaceutical companies will, a lot of times pharmaceutical companies will have biotech entities as well. But when you make a pharmaceutical per se, the strict definition is that pharmaceuticals are either chemically synthesized or they're purified from natural compounds to make the drug. Whereas with biotechnology, you are using a living organism to grow a product or produce a product. Okay, so biotechnology companies are usually growing cells in culture or using live organisms like genetically modified plants or genetically modified animals, and then they're producing the product, which is called a biologic, directly from those organisms. Okay, and biotech companies are not necessarily all pharmaceuticals. Biotechnology companies can be involved in remediation. They can be involved in specialty chemicals production. They can be involved in consumer product production like detergents. Detergents used to clean your clothes have genetically modified enzymes. Okay, so you look at the different revenues here, some of the biggest companies. The biggest biotech company is Amgen, and its revenue in 2012, 2013 was about $14 billion. And then Genentech, which is now a part of Roche Pharmaceuticals. And here are some of the others. And then the pharmaceutical companies like Johnson & Johnson, Pfizer, Roche have much, much larger revenues because they're producing a much wider array of products. To start up a biotechnology company, and I know this firsthand, I had my own startup company in 2000, and we only lasted 2 years. Usually you start with a few employees. My company started with about 7 employees, and you're backed by venture capital investments. And these are large investors that take large retirement funds. They take people's retirement savings and invest them into lucrative companies. And these come from financial institutions or private donors called angel investors. And you want to get this type of investment very quickly because biotechnology is very expensive. And what these donors get in exchange, or these venture capitalists get in exchange, is they'll have ownership in a company that has the possibility of making a very lucrative drug. And so if you have ownership in a company, then you will automatically generate revenue every year. Every year that company sells a drug, then you get a portion of the revenue stream because you own a large part of that company. Okay, so it's very easy to become very wealthy if you invest wisely. A lot of biotechnology startups, even like my own, do not end well. They end up shutting their doors, and so these investors lose their money. Okay, and you have to wait a long time. It takes 10 years to get a product, a pharmaceutical product to the market, and it costs millions of dollars. So you need lots of angel investors, lots of venture capitalists. And then eventually these startup companies, if they're lucrative, they'll be bought by larger biotech companies or by pharmaceutical companies. And when they're bought for a large price by these pharmaceutical companies, the startup entities, the people that own the startup, stand to make a lot of money. Okay, and the value of the company, companies are put up for sale all the time, but the value of the company is how close it is to bringing a drug to market. Say if a company only has 2 years before their product is approved, then that company is going to be worth a lot more than a company that's going to need to get their product approved in 10 years. Okay, and there's also the stock exchange. Company can do what's called go public, and they can sell their stock on the New York Stock Exchange and the NASDAQ Stock Exchange. And when they do that, then they're also set to make a large amount of money because, in effect, they're selling their company to the general public. Different jobs in biotechnology, also research and development, preclinical development, discovery research, bioinformatics, and then also laboratory safety. Operations, once a drug is approved, you need to make a lot of it, or once a different biologic is approved. So you need to develop a process, a large process for making that product where you have large fermenters and purification facilities. And then you have people that monitor the process once it's made to monitor the quality of the product. Okay, and quality control and assurance is always a big deal. Clinical research, looking at how effective a drug is on a clinical population, a population of humans. This is often done with the Food and Drug Administration. So every biotech company has people that are well versed in the Food and Drug Administration regulations, and they interact directly with the FDA. Okay, and then finance. It takes a lot of money to finance biotechnology. And so even people with degrees in biology are going into financial fields because they understand biological sciences, and then they acquire the knowledge of finances either on the job or they might get their master's in business administration. And you can see that the types of jobs, if you want to go into research, you probably want to get a PhD. And where you will be in charge of the laboratory. There's very few jobs that you can get in biotech with a high school diploma, but mostly this would be in biotech production, and it would be a lower paying job. And then to get into research and development, you need, say, at least a bachelor's degree. Good news, you'll have yours soon. And then also a graduate degree. And the best approach to get a job in biotechnology is to get a specialized degree. You'll be working with different specialties. It's always good to have experience in computers. And these days, individuals that are born, in the 1990s and after the year 2000 are very computer savvy. So a lot of these individuals have the type of training already in order to understand computers along with their biological sciences degree. It takes a lot of math, and so there is calculus involved in biotechnology, although again, I will assure you we're not going to do calculus this semester. This is in general. To have a good job, you should have good oral and written communication skills. People that do not communicate well don't get good jobs. Okay, and this biotechnology is a collaborative discipline, so these companies are always looking for people who can collaborate with multiple disciplines. Okay, and it's a very personally rewarding field. I've been in biotechnology for 30 years now, so I'm a walking biotech commercial. Okay, so that ends chapter 1. We'll move on to chapter 2 next. And the next chapter is chapter 2. This is an introduction to genes and genomics. And our objective in this chapter is to look at cell biology. This is going to give you an overview of cell and molecular biology. We'll talk about DNA, the molecule of life, how DNA is structured, replicated, and how genomes work. We'll talk about transcription and translation. And then we'll talk about different mutations of genes. We'll talk about epigenomics and looking at how DNA is packed and folded in nucleosomes. Okay, and so we know that all cells have these structures. They have a plasma membrane, a double layered structure of lipids, two phospholipid bilayers, and also proteins that are embedded into the lipids. And then there's the cytoplasm. There's an inner portion of the cells, regardless of whether the cell is prokaryotic or eukaryotic. And then there are different organelles in eukaryotic cells, not in prokaryotic cells, and these are structures that perform specific functions. Okay, and if you look at prokaryotic cells versus eukaryotic cells, prokaryotic cells include bacteria and archaea. This says archaebacteria, which is synonymous to archaea. Prokaryotic cells have no true nucleus. The DNA is located in the portion of the cell called the nucleoid, which is in the cytoplasm, and they have no organelles. And obviously, prokaryotic cells are smaller than eukaryotic cells. Eukaryotic cells include the kingdoms of protists, fungi, plants, and animals. They can be 10 to 100 microns large. And then you have the true nucleus and you have organelles. Okay, and here's a prototype of prokaryotic cells. You see the nucleoid region, that's where the chromosome is. And inside the cytoplasm, which is inside the plasma membrane, you have ribosomes. And ribosomes are molecules, they're not organelles. I don't want to insult your intelligence, but it's good to review that. And then on the outside of the cell, you have a cell membrane, and you also have a cell wall, but not all prokaryotic cells have cell walls. And then you'll have outside structures like pilus, pili, which are used for conjugation. They're not shown here. A fimbriae, which is helps cells stick together, and then flagella, which are used for locomotion. And then a eukaryotic cell, here's a lot of different organelles, a lot of different structures in eukaryotic cells, including a cytoskeleton. And you see how structured and how varied eukaryotic cells can be. And depending on whether the cell is photosynthetic, it may include chloroplasts. If it's not photosynthetic, then it doesn't include chloroplasts. Okay. And so all cells have a plasma membrane. This is a very fluid, double-layered barrier, and it's made out of phospholipid. Okay. Not only is the plasma membrane used to separate the cell from the environment, but other roles include cell-to-cell adhesion, cell communications, shape, delivery of transport molecules in and outside of the cell, and of course it's a barrier. Inside the plasma membrane is the cytosol. That's this gel-like fluid that's in the cytoplasm. And then eukaryotic cells have organelles where chemical reactions, different processes takes place. And organelles are like organs. Each has a specific function, so each has its own biochemical reactions, and they allow for coordinated chemical reactions in the cell. Okay, and if we go back to where DNA was first discovered, Frederick Miescher, excuse me, coined the term nuclein. It could not be broken down by proteases. And if it was a protein, it would be broken down by proteases, but since DNA is not a protein, it can't be broken down. Miescher discovered that his nuclein had acidic processes or acidic properties, and he renamed it nucleic acid. And obviously, the two types of nucleic acids are DNA and RNA. Okay. In 1928, Frederick Griffith did an important experiment to show that DNA was inherited genetic experiment. He started with two strains of bacteria called Streptococcus pneumoniae, and it turns out that smooth strains of Streptococcus pneumoniae are lethal. Okay, they're lethal to mice. And rough strains, if they don't have a capsule around them, they're called rough. Then they're harmless. So you had S cells and R cells. S cells are surrounded by a capsule, and R cells do not have the capsule. They just have the cell wall, which is exposed. Okay, and Griffith, did the experiment where mice were either injected with the S strain, the R strain, the heat-killed S strain, or a mixture of heat-killed S and live R to see if the mice would survive. Okay, and so when he injected the smooth cells, which are lethal, the mice died. When he injected the rough cells, the mice lived. If he just heat killed the S cells and they were no longer alive, then the mice lived. But if he took the heat killed S cells and he put living rough cells in, then the mice died. And so there was something that was being conferred from the dead smooth cells to the live rough cells, because from the dead mouse he was able to obtain live S cells. So it's not like the S cells came back to life, but the rough cells were able to get some DNA that allowed the rough cells to produce capsules, so they became smooth cells, living smooth cells, and they killed the mice. Okay. What macromolecules made up the capsule? This is usually polysaccharides and proteins. And they heated the S strain because the S strain was the one that was responsible for the killing. Heat killing the rough strain would do nothing. And so what happened in figure D is that the DNA from the smooth cells transformed into the DNA of the rough cells, and the rough cells acquired the ability to produce capsules, which made them lethal. Okay, so in DNA transformation, DNA is taken up by another bacteria. Okay, and in Griffith's experiments, the heat treatment broke open the S cells, and their DNA was released. The living R cells took up the S cell DNA, and it gave the property of being able to form capsules to the rough cells, so they became virulent. And now we use this technology. We can use individual pieces of DNA, and we can transform bacteria all the time. Okay. Other definitive evidence, Oswald Avery, Colin McLeod, and Macklin McCarty. They looked at Griffith's experiments, and then they treated the extracts from the smooth bacterial cells with proteases, RNases, and DNases, and they found out that the Griffith's experiment would work fine with proteases, work fine with RNases, but if you included DNase, then the rough cells were not transformed because the DNA was being degraded. And this gave evidence, because the DNA was being broken down, that DNA was the material that was being transferred from the rough cells to the smooth cells. Okay, so why did extracts treated with proteases still maintain the ability to be transformed? Well, it's because you broke down the protein, and the protein is not what is the vehicle in the transformation, it's the DNA. And again, the RNases didn't work. They didn't block the transformation, because RNA is not being transferred, DNA is being transferred. Okay, and so when we treated the extracts of the smooth cells with DNase, then that would break down the DNA, and so without the DNA, you could not transform the rough cells. Okay, we look at DNA structure. The building block of the DNA is a nucleotide. Each nucleotide contains three parts. It has a pentose sugar called ribose or deoxyribose. There's a phosphate molecule, and then there's a nitrogenous base. The nitrogenous base is interchangeable depending on the nucleotide. Um, some nucleotides contain adenine, thymine, guanine, or cytosine. Okay, and here you can see the different nitrogenous bases. Purines are double ring structures, adenine and guanine. Pyrimidines are single ring structures, cytosine, thymine in DNA, and then uracil in RNA only. And the difference between ribose and deoxyribose is the oxygen on carbon 2. Up here where I'm pointing, there is no oxygen on carbon 2, so we call it deoxyribose. And for plain old ribose, there is an oxygen on carbon 2. Okay. So Rosalind Franklin really elucidated the structure of DNA along with her colleague Maurice Wilkins. They did X-ray crystallography, and they would shoot X-rays directly at the DNA structure and saw that they made an X pattern, and that showed that the DNA was arranged in a helix. And then Watson and Crick used the data from Franklin and Wilkins, and then they also used Chargaff's base pairing rules, and they developed a model of A binding T and G binding C. They really took the best of the other worlds and they combined all this research, and that gave the definitive double helix structure of DNA. Okay, and Watson and Crick published their paper in Nature, the journal Nature, in 1953. So nucleotides are joined together to form long strands of DNA. Each DNA molecule consists of a parent strand and a daughter strand, and the daughter strand is complementary. Okay, and the nucleotides in the individual strands are held together by phosphodiester bonds. That's where the phosphate binds the ribose directly. So I just answered that question. The nucleotide, the binding is between the phosphate group of one nucleotide and the ribose group of the next nucleotide. And each strand has polarity. You always go from the 5' carbon on ribose to the 3' carbon on ribose. Okay, and so the carbons are just, ribose has 5 carbons in it, and you just number the carbons 1, 2, 3, 4, and 5. And the 5' end is where it ends with the 5' carbon on ribose, and the 3' end is where it ends with the 3' carbon on ribose. And then the two strands are held together loosely. The rungs of the ladder are held together where adenine binds thymine and guanine binds cytosine. And these are held together weakly by hydrogen bonds. Hydrogen bonds are not as strong as covalent bonds, so the two strands of DNA can easily be broken apart. Adenine binds thymine with 2 hydrogen bonds. Guanine binds cytosine with 3 hydrogen bonds. Okay, these two strands are anti-parallel, so if one strand goes one direction, 5' to 3', the other strand goes the other direction, 5' to 3'. So they're read in reverse fashion, depending on which strand of DNA is being read. Okay, and DNA, if we untwisted DNA, it would look like a ladder. The rails of the ladder are the two strands, and the rungs of the ladder are the hydrogen bonding between the different bases. Okay, and it looks something like this. And you can see here, here's one strand of DNA, it goes 5' to 3'. There's another strand of DNA, it goes 5' to 3', the opposite direction. And then hydrogen bonding, you have AT has 2 hydrogen bonds, GC has 3 hydrogen bonds. Okay, so the rungs of the ladder are just across here, across, and then the rails of the ladder are the sides. And the backbone or sides, I call them rails. The sides of the ladder are just the individual nucleotide strands. Okay, in terms of genetic structure, what is a gene? A gene is just a sequence of nucleotides that is used to synthesize either a protein or a different type of RNA, like ribosomal RNA. And usually genes are somewhere between 1,000 and 4,000 nucleotides long. I've worked with genes as long as 8,000 nucleotides. And genes influence the cells because they are responsible for the proteins that make up the cells. Genes have traits, okay, and the trait is just some type of characteristic that's conferred directly to the cell. And not all genes are used to produce proteins. There's genes that make transfer RNA, and there's genes that make ribosomal RNA. Okay, and I think I'll stop here. Before we get into chromosome structure, I'll go ahead and stop this video lecture, and this is where we'll start in class on Monday.